Hello, my name is Mrs. Winkler. I am your JCC Edge teacher for Human Biology, course number Bio 106. I'd like to welcome you and thank you for choosing to partake in this hybrid learning experience. We're going to begin with Chapter 1. Shocking, right? Chapter 1 has to do with human biology, science, and society. The chapters that I will be discussing in the lectures correspond with the guided notes that you receive from purchasing your book through the JCC bookstore. In that notebook, or binder rather, is also the book that corresponds with the course. So characteristics of all living things. Lots of times you begin a science course, especially any science course that deals with living things, with the characteristics of living things. First, we'll talk about how living things consist of one or more cells. Unicellular organisms, pretty self-explanatory there. They are composed of one cell. Things that are unicellular would be bacteria or protozoans. These are very simple organisms, so they cannot really carry out complex processes like multicellular organisms. Multicellular organisms, like plants and animals, usually are eukaryotes, which means that they have a true nucleus and membrane-bound organelles. Their DNA is found within their nucleus. Unicellular organisms, like bacteria and protozoans, are prokaryotes, and usually their organelles are not membrane-bound. For example, their DNA is free-floating, as opposed to a multicellular organism or eukaryote, where the DNA is found bound within the nuclear membrane. The more complex an organism becomes, the more chemical processes or reactions that can take place within that cell. Not to say that unicellular organisms are not capable of chemical reactions, because they are. For example, mitochondria carries out cellular respiration, and in plant cells, chloroplast carries out photosynthesis. These two types of processes that occur within these cells are critical to their survival. Living things reproduce, and grow. This is another characteristic that must be demonstrated. The instructions for growth are contained within the DNA. The DNA in a eukaryote is found in the nuclear membrane, within the nucleus or nuclear membrane. In a bacteria, it's free-floating. Many times we think of the DNA as the job description of the cell. It is what tells the cell essentially how to carry out certain functions. The ability to carry out those functions relies heavily on the production of proteins and enzyme synthesis. And as you know, I'm sure enzymes are also protein-based. These proteins that are coded for within the cell are determined based on the arrangement of amino acids. The amino acids are coded based on nucleotide-based pairs and how they pair up and how they go through that coding process, which we will go through at great length as the semester progresses. Some of those nucleotides are adenine, thymine, cytosine, and guanine. The third characteristic that we're going to discuss is homeostasis, a huge buzzword in the living environment. If you're not maintaining homeostasis, which is considered also an internal steady state, also known as dynamic equilibrium, you are in the state of disease. You're sick. Or if it's progressed to a point where you cannot readjust or get back to a state of homeostasis, you are near death. Some examples that I have here that allow for homeostasis is normal blood pH and sweating. Living things require energy and raw materials. Your metabolism. The metabolism is a physical and chemical process that transforms energy and molecules to sustain life. We know that in our bodies, mitochondria carry out cellular respiration, and if we do not do that, we can't get up in the morning, we can't breathe, we can't eat. We would be in not a state of homeostasis. In plant cells, plants need to carry out photosynthesis in order to produce glucose and oxygen in order to survive and live. Producers, as I said, trap this energy. What is the energy? Comes from Mr. Sun up here to carry out photosynthesis. Photosynthesis is carried out in the organelle known as a chloroplast. The chloroplast contains chlorophyll, and this chlorophyll is where photosynthesis is taking place. Consumers, like yourself, are animals that feed on stored energy in the plants. So you can see how we have a relationship here. Producers use that CO2 to push through photosynthesis. At the end of photosynthesis, we have glucose and oxygen that is made. That glucose and oxygen is then utilized by your cells to create energy in the form of ATP through cellular respiration in your mitochondria. Decomposers are essential. If it wasn't for the decomposers breaking down the dead and dying materials that are found on this earth, it would pile up and we'd have a boatload of methane choking out our available oxygen. Bacteria and fungi are responsible for this breakdown for the most part, and they do not recycle energy. Let me repeat that. They do not recycle energy. They recycle the raw materials utilized for other processes. Number 5, living things sense and respond to specific changes in their environment. So this essentially means that you are able to detect when things have changed. Our sensory organs, are found in the central nervous system. So that would be your brain, your spinal cord. You have two, a parasympathetic and a sympathetic nervous systems. So how does that work? Okay, so let's talk about that. For example, you're in a dark alley, you hear a loud crack, and all of a sudden your body responds. You have the whole fight or flight response. You're either going to stand there and stick around and fight it out, or you're going to run like heck to get to safety. So if you're standing there, your endocrine system is also involved because it's going to be releasing adrenaline. That adrenaline will charge you, will make your heart beat faster, cause you to breathe faster, to produce more ATP, getting the nutrients and the oxygen to the cells to produce ATP, especially in your muscles, to make them be able to work, to get you out of there or protect yourself. Same as if you smell smoke or you are threatened, that whole reaction takes place. Plants, on the other hand, go through what's called phototropism or gravitropism. Phototropism, if you see sometimes you have plants that kind of are bending towards the light. Gravitropism would be the gravity, or they're looking for the water, so their plants are sending their roots downward. Populations of things evolve. We have to change. We have to be ever changing in order to survive. Changes don't happen quickly. This is a slow change over time, and characteristically is driven by environmental conditions. These changes take place, as I said, over many generations, and the DNA, there are different populations of things that have differences in their DNA. These differences, if they're beneficial, are bred in and become a part of the fabric of that new species or the changed species. What does that give us? Diversity. Diversity is huge in the success of a species. Gene mutations are not all bad. Many of them are very good, that result in structural and functional changes that enable us to survive through different stresses. So, a key evolutionary idea is that individuals, in fact, vary. Individuals don't evolve. Populations evolve. Not involve, evolve. Let's try that again. Evolution occurs where there is change in the genetic makeup of a population. So, for example, here's the old-timey giraffe adage here, where the giraffes, as the leaves that were disappearing in the lower shrubbery, those giraffes that could reach up to the higher trees where there was food, they were able to eat. They were able to maintain homeostasis and remain healthy. So that gene for a longer neck was passed on through reproduction. What happened? The ones that couldn't get food began to have the homeostasis be disrupted, died off. So, as I already said, variation comes from genetic differences. Another variation would be the peppered moth. One that is very common would be known as the rock pocket mouse. That's another example that we see today as we speak. Mutations are things that are heritable, so you can inherit them. There are changes in your DNA, and that is where your new gene forms. So now let's go on to talk about genetic variation and natural selection. Individual variation exists within a species. We know that. There are different types of traits. There are adaptive traits. Some forms of a trait are more advantageous to another. What does that mean? That means they improve your chances of surviving. So let me go back to that rock pocket mouse. That rock pocket mouse in the Sonoran Desert. There was a volcanic eruption, and with that volcanic eruption, the lava flowed out and hardened and caused the rocks to change color. They were a grayish, whitish color, and now they are a darker color. So this rock pocket mouse had the mutation to have a darker coloration. Who survives? And characteristically, what they're called is the Snickers bar of the Sonoran Desert, because they're real small, and birds of prey eat them. Many different things eat them, but birds of prey, they're easy to spot if they're white on a dark colored rock. But the darker ones are able to camouflage themselves and survive. Another couple of examples would be a lack of the CCR5 receptor for HIV, or more common right now would be the antibiotic resistance in bacteria such as MRSA, and you can see that on page 18 in your book under the Health Watch. And I'm checking the page as I'm doing this because there was some changes in book numbers, but I'm quite certain, yeah. Okay? Yep, we should be good. All right, so human features. Increased dexterity. Everybody knows about the opposable thumbs and the relationship there. Bipedalism, the ability to walk on two legs. Your large brain relative to your size. That would be, we'd be speaking of your cerebrum. Communication is the last one. So living things are now classified according to their characteristics. Okay? So, when we're talking about that, okay, there are 5 kingdoms. The monarans, the protists, the fungi, the plants, and the animals. These are the kingdoms. So there's 5. Domains, there are 3. You have bacteria, which you're familiar with. They are unicellular. A great example, which you've heard spoken about, is E. coli. E. coli can be very deadly when it is found in raw or uncooked meat, on unwashed spinach. RK is bacteria living in harsh habitats such as where there's no light, in very harsh, high salt conditions, or in dark chemical environments where there's like a lot of methane. And here you would find like blue-green algae. Eukarya is single cell and multicellular. So you have protists, fungi, plants, and animals. So we have our classification. You have the domain, kingdom, phylum, class, order, family, genus, species. I'm sure that you have learned this in previous subjects such as AP Bio or Living Environment. And here we go through just a classification of humans. We are a part of the domain Eukarya, the kingdom Animalia, the phylum Chordata, class Mammalia. We are the order Primates, family Hominidae, and our genus and species is Homo sapiens. So now let's move on to the levels of biological organization. So this is in your book on page 10 and page 11. And here you'll see we are organized from smallest to biggest. So you have the atoms and molecules, and you have groups of those make up your cells. Then you have your tissues, your organs, organ systems, organism, population, community, ecosystem, and biosphere. So for example, groups of cells make tissues. So one of the stumpers is always blood. Blood is actually a tissue because it is made up of groups of cells. What groups of cells are found in blood? Red blood cells, white blood cells, and white plasma, platelets. There's many things. So if we were to talk more specifically about this, and on page 11 you will see that this goes into greater detail. So you have your cell, which is your smallest living unit, and then here I want you to begin to think about your cell theory. Tissues are groups of cells. I already talked about that. Organs are tissues combined. Groups of tissues make up an organ. So like the stomach is an organ. Your stomach, your intestine, your large intestine are all organs that make up your digestive system. So that's an organ system. All your organ systems together, so your skeletal, muscular, respiratory, cardiovascular, and so on, make up the individual. Groups of individuals make up a population. They're same kind, same place. Groups of populations make up a community. Groups of communities make up an ecosystem. And finally, you have your biosphere. We're going to move on to the next piece, which has to do with scientific reasoning. Now, again, this is something that everybody has had in the past that has to do with the scientific method. Many, many science courses begin their years this way. So, let's talk about inductive reasoning. There are facts that are accumulated through observation until the weight of evidence allows a logical statement. This is essentially your conclusion. And we'll be talking about this year in the end pieces and how do we get to executing an experiment and being able to discuss it very well to convey what we've learned. When you begin to brainstorm, this brainstorming process that takes place within amongst individuals allows us to come up with a question or something that we want to answer. So there, I think one of the things that happens with kids is they don't realize how they can apply the scientific method to many questions or problems that they have within their life. And at this rate, let me go back here to hypotheses. We're past the if, then, and because stage. So your hypotheses must always include your independent variables and content. But you should be able to move away from the if, then, and because format of a hypothesis. Next, we have deductive reasoning. This is generally a statement that leads logically to one or more conclusions used to make a prediction that support your hypothesis. You can begin with that if-then, but you will not write it as an if-then. You will elevate yourselves to a higher level. So, the scientific method. How do you begin? Observation. New observations are made. Raising questions. You check for previous data. You check to see, did anybody else ever do this experiment before? If so, what did they discover? Then you develop your hypotheses, you formulate your question. It should be testable. It can't just be a question that you can't do anything about. Many times it's referred to as an educated guess. So then you make a prediction. Again, do not use the if and then. Make sure you incorporate here your independent variable. Okay? Should state the results expected if the hypothesis is correct. One important piece here, though, you will never, ever, ever tell me whether you were correct or not. You will give reasoning. You will make a claim. You will state your claim. This is what I determined. Then you will utilize the evidence that you gathered based on your experiment. Then you will give reasoning. But to say my hypothesis is correct is very elementary. So, next you conduct your experiment. You make observations. It needs to be a controlled experiment only testing one variable at a time. You will repeat and modify your hypothesis as needed based on your findings. Then you will analyze results and report your conclusions. If, when you do this experiment over and over again, you get the same findings, you then can proceed to develop a theory and possibly a principle. So let's look at experimental design. Variables, what are they? They are factors that may vary during an experiment. There's two types, an independent and a dependent variable. Independent variables are things that you change. Dependent variables are data that are collected based on the experiment you conducted. A control group is that which is a standard used for comparison. Experimental group tests the control variable. Examples. You might want an example here listed, how nitrogen content affects the growth of grass, or how a drug may affect blood pressure. Blind experiment and double-blind experiments. Ask yourself what these are. You never know when it may come up again. So your scientific method, let's just go over this again. What makes it valid? Large groups of or large test subjects or large populations or subjects are tested. Randomly divide. You have a group 1 and a group 2. You have an experimental group receives the treatment. The control group receives the placebo. You make observations. You collect your data. And then you write your conclusions. Clinical trials are studies that are designed and conducted. Usually an example I should say would be like with pharmaceutical companies. For example, a drug trial, the control group receives a placebo. Animal testing, this might take place on. Phase 1 is usually less than 100. Phase 2 is hundreds with the disease. 3 is thousands. Human guinea pigs, has this happened? Unfortunately, yes. One of the most popular human guinea pig studies was the Tuskegee syphilis study done during 1932 to 1972. 500 poor black rural men from Tuskegee, Alabama received syphilis. They were never treated from it. They were just observed to see what happened to them. They were never even told. They were told they were being treated for bad blood. In the 50s, they had volunteers that were infected by gonorrhea. Some critical thinking. This is my favorite piece, personally. Critical thinking is really and truly, in my opinion, that which leads to the greatest of ideas and the greatest of possibilities of how things could turn out. You want to analyze information here to make good decisions. Utilize the content that you have to help you make good decisions. It's important to note that when you are using critical thinking skills, you use reliable sources to educate yourself. Question everything. That's the most beneficial tool you have in your scientist's tool belt. It's the ability to question. It's that ability that gives you the drive to find answers. Scientific evidence versus anecdotal. Check your data. It should not be hearsay or testimonial. Make sure the interpretation of the data is correct. This is a big problem if large groups of data have not been collected. Sometimes the data that you're looking at, if it's a small data set, can be misconstrued. Correlation versus causation. For example, there is a huge controversy between autism and vaccines, and in fact, that is the reason why we did what we did with our discussion piece. So I'm going to end here with Chapter 2. This is the first of your lecture series. Chapter 2 will arrive on your doorstep shortly. Thank you very much. Have a great day.